So here I'm going to work through problems in problem set C. The first question in problem set C says, order the following molecules from the highest oxidation state to the lowest oxidation state. So let's consider these molecules. First of all, we have ethanol, CH3OH. We have carbonic acid, which is CO2H. We have formaldehyde, which is CH2O. And finally, we have methane, which is CH4. Well, these are actually fairly simple to do. This is an acid. This is an aldehyde. This is an alcohol. And this is just simply a hydrocarbon. If we compare them at oxidation states, we remember from our organic chemistry that the lowest oxidation state, that is the most reduced compounds, will have no oxygens. And so each of these has one carbon. We can readily compare them. This one carbon compound has no hydrogens, has no oxygens. Therefore, it will be the lowest oxidation state. The next lowest will be this guy here, the alcohol. Okay, so we'll call this one 1. We'll call this one 2 in terms of the lowest. The third will be the aldehyde here, and the fourth will be the acid. And if I had included carbon dioxide in there, CO2, then I would have put that actually as number 5, and it would have been the most oxidized of all of those molecules. Let's consider the situation we have here, where we have two different molecules, an R attached to a sulfhydryl group and an R attached to a hydroxyl group. And we remember that electronegativity is a measure of a given atom's affinity for electrons. So if we look, for example, at a sulfhydryl here, we could think of that sulfhydryl as something like this. And when we think about this hydroxyl, we can think of the hydroxyl as having electrons that look something like that. Atoms that have higher electronegativities will pull electrons closer to themselves than will atoms with lower electronegativities. So for example, oxygen will pull electrons closer to it than hydrogen will, and so the electrons will go that direction. Sulfur has a higher electronegativity than hydrogen and will pull electrons closer there. Well, which one is pulling stronger really is the question here, and it turns out that, of course, oxygen is pulling them stronger. And so oxygen will have what we call a partial negative charge, and sulfur will have a partial negative charge. And the magnitude of that partial negative charge will be a function of how close the electrons are. Well, in both cases, the electronegativity of hydrogen is the same. So since oxygen has a greater electronegativity than does sulfur, oxygen will pull those electrons closer to itself. It will have a greater positive, charge, partial charge than will sulfur, and therefore will make stronger hydrogen bonds. Can't write. Because there'll be a greater charge difference between these two than there will be between these two. The third problem in problem set C says that porin is a protein embedded in a membrane. So we can think of a lipid bilayer as having these little tails sticking down and in, like I've described here. And porin is a protein that's embedded in this lipid bilayer. It might look something like that. It says, predict how the orientation of its amino acids will be different from those of a protein found in the aqueous environment of the cytoplasm. So imagine we have two proteins. We've got porin, which is sitting here, and elsewhere in the cell, we have another protein, which I'm going to draw with some jagged lines looking like that, that's dissolved here. Well, these two proteins are in very different kinds of environments. The porin, which is located in this case, is embedded in a region of the membrane that is very non-polar. Whereas this guy here is in the aqueous environment of the cell, which is very polar. Well, what happens when we have polar molecules interacting with nonpolar molecules? Well, they tend to avoid each other. Water tends to avoid oil as a consequence. You may remember when I talked in class about the folding of a protein, as I said that proteins that are dissolved in water, like this guy is right here, will tend to orient their amino acid side chains so that they are on the inside of the protein. Okay, so we'll put dots here to indicate nonpolar amino acids. All right, so that they're on the inside of the protein so they can interact with each other and avoid interacting with water, where I'm going to put the polar amino acids. So the polar amino acids in this case are oriented on the outside, interacting with water, and the nonpolar ones are on the inside, interacting with each other. Well, that's a very different circumstance than we have here with porin, because with porin, the things that are on the outside here, okay, are oriented such that they're touching all this nonpolar stuff right here. We're going to see on porin, in many cases on the outside, we're going to see some of those nonpolar amino acid side chains interacting with the nonpolar portions of the membrane. Only out here will we see polar amino acids, only where there's water touching them, okay? So the orientation of amino acids in porin will be very different than they are for an enzyme or protein that's dissolved in the cytoplasm of the cell due to the avoidance of the nonpolar side chains for the aqueous environment of the cell.